For Microbe TV, this is Immune, episode 80, recorded on May 14th, 2024. I'm Stephanie Langle, and you are listening to the podcast about the body's defenders against disease. Joining me today from Ithaca, New York, Cindy Leifer.
Hi, welcome everybody.
Great to have you. We saw each other very recently.
We did. We were all in Chicago.
Which was lovely.
Yeah.
And joining from Madison, New Jersey, Brianne Barker.
Hi, great to be here and great to see you both again.
Yes. Yeah, we were in the Windy City. We did some recordings.
Yeah, we'll see when they come up.
Yeah, I'm excited. We had a great group of people.
Oh, it was
very
amazing.
Responsive.
Much fun.
So much fun. From when you sent that tweet out, that X out, Cindy.
Yeah.
We were able to get them all scheduled. Boom. I mean, with Karen, we have a wonderful production person, an IT person who helps us with all this, of course.
Yeah.
But great. Yeah, that was lovely. And today we're coming together because we want to go through, we're going to do a round robin of papers. We each have prepped one paper, and then we have another little snippet that we all agree that we looked at our second papers, but we may not have gone through them deeply. So if we have time, we'll do that as well.
Yeah.
So the first on the list, Cindy, do you want to talk about your paper?
Yeah, and before I do, I just want to make sure that everybody knows that if you enjoy what we do, including the papers we're going to go over, please consider supporting all of the science shows, including Immune, at Microbe TV. Which you can find at microbe.tv/contribute. So my first paper is one that got a lot of press. And I think is quite controversial and will be interesting to discuss. So the title of the paper is Intranasal Neomycin evokes broad spectrum antiviral immunity in the upper respiratory tract. Now, you might be thinking, well, wait, neomycin is an antibiotic, and we're talking about antiviral immunity. Everyone told me since the dawn of time, antibiotics do not treat viral infections, right? Right. So this paper was published just a couple of weeks ago on April 22nd in PNAS, or the Proceedings of the National Academy of Sciences, to which Akiko Iwasaki, the senior author on this paper, was inducted into the National Academy and can contribute papers to this journal. So we need to think about that. It was reviewed, but it was contributed by Akiko. Not that Akiko is a bad scientist, but we can talk about this paper. The first author is Tianyang Mao. And there are two other, actually like 25 other authors, but those are the main ones involved in the study. So what came before this was that this group, so Akiko's group, had previously used neomycin in the vaginal tract and showed that vaginal application of the neomycin, which is an antibiotic, drove an interferon type of response that was dependent on TLR3, and that interferon response was protective against some vaginal viruses. So the purpose of this paper was to say, okay, well, we've got respiratory viruses. They're clearly an important global problem, as we all might know from the case of the COVID-19 pandemic. And the idea is, like, if there's anything we could repurpose, so existing approved drugs that could be repurposed, it could be a rapid and cheap way to treat diseases. And they argue this is especially important in the developing world. So this study, they put intranasal delivery of neomycin. And so if you don't know much about neomycin, it is an antibiotic. It falls into the category of aminoglycosides. And it does, based on what they've previously shown in the vagina, they also showed now, looking in the lungs, that it induces expression of interferon-stimulated genes in the nasal mucosa. And they went on to do a lot of different studies, but one showed that this was completely independent of the microbiota. And that's sort of an important thing, because the microbiota make metabolites, and a lot of different things have been connected to what types of microbes you have in your gut, or whether you have them or not, or what kind of metabolites they make and how they're communicating with immune cells or the brain. So it's important that they were able to show that this is independent of microbiota. With respiratory viruses like SARS-CoV-2, they usually start in the upper respiratory tract and then they go down into the lower respiratory tract. And the ways we normally think about fighting them are we either target the viral replication strategies, so entry, replication, or exit, or we target the host. And this group actually is thinking about targeting the host, so inducing these antiviral defense genes by just squirting some neomycin in the nose, driving the interferon response genes. And therefore, if you had these higher levels of interferon response genes, then when you get exposed to a microbe such as SARS-CoV-2 in the model system that they're using, you would be protected. So the first problem with this paper, and I don't want to tear it apart because it's interesting, but there's really not much new about this. It got a huge amount of press because people are thinking, oh, I can just take some neomycin or just some, now I'm forgetting what you call it, the neosporin, and just wipe it into my nose and I'm going to be protected from viral infections, right? So it's
Right.
so simple, right? So it got a lot of press, but the thing is they did this already in the vaginal mucosa with a different virus, and they already showed a mechanism, and now they're just doing it in the upper airway in the nasal mucosa, and then exposing to the virus in the nasal mucosa and saying, yep, works there too.
Yeah.
So-
Well, I think there are a couple of questions just coming out of these experiments that you've mentioned. You mentioned that they do this, but the first question is, well, how long does it last?
Yeah.
In theory, if you're turning on interferon responses, if you're turning on interferon stimulated genes with this neomycin, you're probably not turning them on for months or years or something like that. You're getting a very short-term protection against
Correct.
viral infection. And so it could be something that could be useful if you know that later today you are going to be exposed to a virus.
Maybe if you're about to go on a plane and you wear your mask, but you also want to stick a little neosporin up your nose.
Yeah.
Sure.
So I think that the timing thing is weird. And I think that the other thing that's important to remember is that these are mice.
Yes.
And we are unable to ask the mice how they feel
About
this. And in particular, if you're inducing a big interferon response, the mice may not feel the greatest.
feels so good, right?
They may have some negative symptoms, as we all have when we're having a large interferon response. And so,
Yeah, but I guess I guess this has to do with threshold. And this was the question I have with this paper about dose responses. It was, if I remember, they didn't do dose response studies.
Nope.
Because to your point, Brianne, I mean, a large interferon response is not good. People do not feel well. But I just can't imagine that neosporin, or I don't know what am I thinking of the threshold is, and then how that correlates to the level of interferon, and is that protective, right, for how long?
Right. So I think there's a couple of things here. One is that it definitely goes up and goes down, right? And the protection is going to be there for a certain amount of time. In this model, they kept dosing them every day. And even when they got to human studies, and we can talk about that, it was every day, right? We can't, that's not practical. We're never going to be doing that. The other thing to keep in mind is that there have been delivery of interferon as a therapy, and it's not very well tolerated. Right. So when you have chronic viral infections, they thought, hey, well, why don't we give them a lot of extra interferon and maybe that'll clear the virus? And it really, in fact, it doesn't feel very good when people have shots of a lot of interferon. So like you said, Brianne, we can't ask the mice how they feel.
right.
But
I mean, we can ask them. I just don't think they'll respond.
Squeak, squeak. The other thing is that the delivery mechanism, right? Okay, so we're all saying, okay, let's put neosporin in your nose, which does have neomycin in it. I looked this up, and I'll tell you that in a second. But for this study, what they did was they put 10 microliters, which doesn't seem like a lot, but mouse noses are really tiny. They're tiny, tiny.
So tiny. Yeah.
So it is liquid, 10 microliters, that has 2 milligrams of neomycin sulfate. That's a lot of neomycin in a small volume in a little teeny, teeny, tiny nose. It's not the same as Neosporin.
Yeah, have you, and did you scale that up? Like how much would I need to squirt in my nose to get?
So I didn't do the scale up. You guys could do
Sure.
the math in your head, but what I did do was look up Neosporin. So Neosporin, which, is the brand name of the little gel that you put on your cuts, right? So it has neomycin, it has polymyxin B, and it has bacitracin. And the neomycin amount is 3.5 milligrams per gram of gel. Now, I did not go so far as to go get a little tube of neomycin and measure out how much a gram is, but I'm suspecting that's a lot of gel.
I would say so.
I would imagine that you're like stuffing your entire nasal cavity with
Packing it in.
neomycin.
Yeah, so I have done intranasal inoculations in guinea pigs.
Mhm.
And I was just looking at the methods because I had a question that they really do not address here, which is that at least in the situation where we were doing this, we would really have a problem with the animals sneezing the stuff out that we dripped in their nose.
Yep.
And it was a big deal to think about how to potentially anesthetize them or how we dealt with the sneezing. And they don't mention anything about that. They don't mention anesthetizing the mice. They don't
I think they anesthetize them, but yeah, 'cause they're not going to enjoy having you squirt the liquid up their nose when they're awake.
It looks
I
like
would
they
think.
anesthetize for viral infection, but they don't mention it for the neomycin treatment.
I'm guessing they did. But
And to
I
that
guess,
point,
yeah.
So when we were doing our SARS-CoV-2 in hamster studies with, we were vaccinating before and challenging with a liquid, and it doesn't just stay in the nose. At least, so this is
Oh, no,
hamsters,
they
but I'm
breathe it,
assuming,
right?
Yeah.
and we had to do dye studies to see, okay, well, what other lymph nodes is it accessing? So that's another point, and I don't remember, Cindy, correct me. I don't think they show you with a dye where else is this going.
Nope.
Yeah, they actually say that it was. Oh, actually, no. They are anesthetized. I'm seeing that now.
Yeah, I thought so.
But they administer it drop wise into the nasal cavity using a pipette tip. So I mean,
it's not so easy to drop wise 10
Yeah.
microliters. That's like 1.5
I'm trying to
drops.
imagine getting like drop wise of microliters into the nose and the mouse just being chill about it.
I don't know.
But yeah, I mean, I think you're right. The delivery aspect, and they mentioned that in their limitations, that this is not, they've not really tested this in humans, but the scaling up. But it's interesting if we can think about, okay, so now we know this, what are the mechanisms that lead to this induction, and is there a way that we can harness this? But it's always the problem with viruses and antiviral treatment in general. When you get people to the hospital, their viral replication has already peaked, likely.
Mm-hmm.
And it's rare that you're going to be taking an antiviral daily. That's why we get vaccinated. So we have those memory B cells ready to go, and T cells.
Yep.
So, I always think about antivirals in this way. And Cindy, did they do the infection and then they tried to treat?
Yes.
Okay. Did it also knock
They
it down?
did a post-exposure test, but it was 4 hours.
Right. Okay. So not like a couple days and you're going to the hospital because you have respiratory symptoms.
So it's not super great. They did do 4 hours. So it wasn't just before, but they did. But I mean, that's not really post. That's like, okay. Yes.
Yeah, I also do not tend
Is it
to know
replicating?
4 hours later, oh my gosh, I just got a virus.
Yeah, maybe if you're rapid testing every day or something. But yeah, titers maybe would not have even peaked by then. So yeah, I think that's a challenge with mice studies, with antiviral studies. The timing really matters.
Yeah.
Yeah.
I think a couple of other important things to mention about this paper is that they did test, like, for example, they used interferon knockout mice, and they showed that it isn't simply the induction of interferon that induces the interferon-stimulated genes. So that's an important thing, because if the only thing that it was doing was inducing interferon, then just giving interferon is probably the same or better, right? So it's somehow directly inducing those interferon-stimulated genes. Now, they didn't go into the mechanism as much as they did in the prior paper when they delivered it vaginally and showed that there was a TLR3-dependent activity. What else did I want to mention? So they the model that they used in this study to show that this was protective was SARS-CoV-2 in the K18 transgenic mice with basically a lethal version of the infection model.
Right.
But they did also use influenza A, and just for if Vincent isn't here today, but to let him know, they did do a plaque assay to show that they reduced viral load. But I mean,
always good to do a plaque assay, right?
But I mean, this is almost like a duh. Of course, if you induce interferon and interferon-stimulated genes, you're going to reduce viruses and viral replication. A cute thing that they did was they did use the Syrian hamster model, and what they showed was that in that model, if they co-housed uninfected animals with infected animals that had been pre-treated with neomycin, it prevented the transfer. So
knock down viral replication,
again,
less to transmit. Yeah. Okay.
Again, right. So I think that what got people really excited about this study in the end is that they did do humans. They did do a randomized, double-blind, placebo-controlled study with a few individuals who took Neosporin or Vaseline and stuffed it in their noses, self-administered with a cotton swab. How accurate the delivery is, the volume and all of that, I didn't see exactly how that, the details of that and how that happened, but let's assume they did it correctly and everything. They did see that there was no protection because nobody got infected as far as I remember reading it. But what they did was a nasal brush sample. Now, imagine being in this study. I'm taking some neomycin. I'm wiping it in my nose with a little cotton swab. And then the next day they go in and they brush the inside of my nose and scrape off epithelial cells. And then I go put Neosporin back on again. And then they scrape it off again. It's what they can do, but it is a very crude way of looking at things. But what they were able to show is in the nasal brushings, they could see an increase when they looked for interferon-stimulated genes. They saw them go up more in the neomycin group or the Neosporin group versus the Vaseline group. So again, duh. If you have something induces it and you put it in the nose and induces it, okay. So what do I don't know.
Yeah. And sorry if I didn't say the right thing before. I said one of their self-limitations was that they didn't show efficacy against a viral infection in humans, but you're right. They did do this clinical trial.
Yeah, they did do the clinical trial for looking for interferon-stimulated genes.
Right.
So the presumption is if you induce them, then you would be protected. But for how long?
How much?
Yeah.
And then do we want to be administering antibiotics more? I mean, don't we have
so this is a big problem.
Antibiotic stewardship issue.
This is the problem that I think with the media making such a big deal about this particular paper and the risk is that people are going to start trying to use antibiotics to treat viral infections, right? Because it shows that you can do it. And if you don't understand which antibiotic it is and the mechanism of action that it's working, it's not any antibiotic. You don't go take some, I don't know,
penicillin.
pills that you saved from the last time you were sick, right? And take them and expect them to prevent a viral infection. This was a very, very specific way of doing it. Delivery of one particular antibiotic that happens to, they didn't show it here, but they have in their other mouse studies, trigger TLR3, which promotes a very strong interferon and interferon-stimulated gene response, which now if you have that on board, it's basically your shield is up, and so it's much harder for the virus to get a foothold. And it prevents the infection in a mouse nose that was oversaturated with an antibiotic. This is completely different than just taking a random antibiotic for treating a viral infection. And it's a risk because we cannot have poor antibiotic stewardship.
Yeah, or even
because
the
this
messaging,
relates
to your point.
Yeah, I mean, this
Yeah.
leads to antibiotic resistance. We know that. When you're
Yeah.
using antibiotics, when you shouldn't be using them, you have an increased potential to generate resistance to those antibiotics, and then we will have fewer of them. And that is what we have against most bacterial infections
Yeah.
to
So
protect us.
one, I guess, positive of this is that maybe, and they don't really show us this, maybe these ISGs are only being produced in the nose. And so maybe we're not going to get the same kind of systemic interferon production and the systemic feeling bad. So maybe the sort of local
Yeah, I would
aspect
agree
could
with
be
that.
useful.
Right.
But I also would like to know, I do not particularly want to encourage the growth of neomycin resistant microorganisms in my respiratory tract.
No.
But I also am not sure what happens if I'm putting Neosporin in my nose frequently. I take an inhaled allergy drug, and so for some reason I'm imagining this being similar to my inhaled allergy drug that I take in the mornings. You know, inhaling my Neosporin every morning to protect me from viruses. What's that going to do to my nasal microbiome? What's that going to do to my protection against infections with nasal bacteria? Am I now
Right.
going to have big changes in what's going on with bacterial infections because of this?
Yeah, I think all good points. And thinking about, okay, if you were the graduate student, postdoc, whoever's doing this, to be fair, they took previous work that they had done that showed an effect, and they said, hey, I want to know if this works. Is this do I see the same thing in the respiratory tract? They did
Fair.
quality work with controls. They
Yep.
wrote it up and they published it. It's not their fault that the media is responding. And I can imagine if I'm the grad student, I'm like, I have no way of telling them. I mean, I can get on Twitter and say, no, these are the limitations, but maybe our reaction to this paper is a certain way because the media and not the paper. Do you feel, Cindy, like they were overselling it, or were they just telling you the results?
Mostly telling the results. I mean, they
Okay,
did talk
okay.
about a little bit of the limitations in the end.
Sure.
Yeah.
Sure.
Well, that's interesting because I was, at AAI, I was talking to somebody, and I can't remember who it was, that we were talking about how we are all aware of some of the limitations of our papers, but we're very reluctant to put that into the paper because we don't want to tell the reviewers the limitations of the paper,
100%,
right?
100%, because then it's going to be another 3 months of experiments that are none needed.
And there's always limitations, right? There's not going to be any paper that has zero limitations.
Right.
But the idea that I thought was really intriguing was once the paper is accepted, then you write a limitation section that gets tacked on the end. So it really tempers and puts things in perspective, but it doesn't deflate or take away from the message and the review of the article. And I thought that was a really interesting idea.
Yeah, I like that. The other thing that I would do is we have this platform, so this is kind of a privileged take, but I would just immediately record a podcast with myself and my team and just talk about the paper so you can get ahead of the story so that you can be on record saying we know the limitations. I don't know. I think there's a way that you can go out and message. I was seeing on Twitter there's some similar thing going on where there was this vitamin D and cancer paper, and the lead authors are very much telling just what the data says, but there's a lot of grifters, especially nutrition supplement people who are like, vitamin D treats cancer, and the lead author's like, no, like that's not what we're showing. So it's hard. Once the message is out there and skewed, it's hard to reel that back in. And I don't necessarily think that's the fault of the author because we write it scientifically. You're right. So the reviewers don't, ding us for that. So I think part of the nuance for sure.
Definitely.
Yeah.
But I think the takeaway is that, if you induce interferon and interferon stimulated genes in your nose, it might prevent viral infection.
Okay.
But I thought one last thing I think is really worth mentioning. And that was that their neomycin treatment also induced the interferon-stimulated genes in germ-free mice. And the reason why I think that's interesting is because it is sometimes part of the cocktail of the antibiotic mixture that people use to deplete microbiota.
Interesting.
So when you're looking at what the effects are after antibacterial treatment and depletion of microbiota, could there be effects that are not actually attributable to the microbiota, but could be attributable to the antibiotic effect on the host?
Right. TLR3, innate immune activation. Correct. It seems like that is what they're showing us, which is...
Right. So I think that there is something to consider there. I'm not suggesting that every, microbiota depletion paper that has ever been published is now crap, but I think it's worth revisiting and asking whether some of the effects that people have seen when they've depleted microbiota with complex mixtures of antibiotics that might contain things, they might be it might be a combination effects of direct on the host as well as on the microbiota. And it's something to consider.
I think that's a really interesting point.
And then deriving them in a germ-free facility. So raising the pups, and that would be then what you would have to do to prove that it's not an antibiotic-mediated.
Well, comes with its own complications, because when you have germ-free mice, they have their own immunodeficiencies until they're exposed to microbes and such. So, I think you have to look at things holistically with lots of different models, understand the caveats of each model.
Sure.
Is really the only thing you can do.
Right.
Cool. Very cool. So I have a paper called Sexual Dimorphism in Skin Immunity is Mediated by an Androgen ILC2 Dendritic Cell Axis. This is from Yasmin Belkade's lab, who is a pretty prominent immunologist. You can see we all shook our heads.
Yep.
And the first
she's
author...
going to go lead the Pasteur Institute.
Yes.
Correct?
Yep. Yeah, she's leaving.
I mean, I think she's retaining her lab at the NIH, but then is also going to,
Yes. Well, this is her work that she's still done at the NIH, and the first author is named Liang Qi. And they do a nice job kind of at the beginning of this paper talking about a bunch of different pieces of background information that I first wanted to sort of mention. Largely, they talk a lot about differences in immune responses between males and females. And those immune responses have been observed in a number of settings and lead to things like differences in amounts of autoimmune disease, differences in outcome of infection, and many, many other things. And there have been a number of different things that have been thought of as the mechanism here. One of them has to do with sort of gene dose issues, whether a gene is on the X chromosome and females have 2 copies of it. In particular, there are TLRs on the X chromosome that seem to escape X chromosome inactivation. And so it may be a genetic thing of having extra copies of that TLR. Alternatively, there may be some role for different hormones in actually acting on immune cells. And a researcher named Sabra Klein has done a lot of really fabulous work on all of this. But one of the things that I hadn't really thought about until I read this paper was that much of that work is done, as is much of immunology, looking sort of systemically at the immune response. And here the authors wanted to look at a specific tissue and to see if this disparity was seen in all of the tissues they examined, or if it was seen more so in some tissues as opposed to others. And this group has done a lot of really great work on sort of tissue immunity, particularly skin immunity. And in fact, they found, they cite in the intro that when they actually looked at 44 different human tissues, and the number of genes that were differentially regulated between men and women, skin was particularly different. Which I was like, oh, that's cool. So
very cool.
They really wanted to kind of determine if there were differences between male and female mice in immune responses in tissues, as opposed to looking sort of systemically in the blood. And they first just looked at T cell numbers in different organs, and at steady state, the female mice had more of a whole bunch of T cell subsets in the lung and in the skin, but not in the draining lymph node or in the small intestine. So lung and skin seem to be really unique and have way more of all sorts of these T cell subsets in the female mice.
So remember we did the number of immune cells and the frequency of immune cells in the tissue, and we talked about male versus female. Yeah.
Yeah, absolutely. So based on this, the numbers are pretty different. So we'd have to go back to those papers, particularly in, like I said, the skin and lung, but not in the intestine or in the draining lymph node. And they looked to see whether this is dependent on the microbiota. And they found that and so they used germ-free mice like we were just talking about. And the male and female difference persisted in the skin in germ-free mice, but not in the lung. And so they said, there's something weird going on in the skin in male versus female mice with T cells. And so they only looked at the skin in the rest of the paper.
And when they looked in the skin at T cells, were they only looking at alpha beta T cells? They also looked at ILC2s, but did they look at gamma deltas?
They looked at gamma deltas a bit. So they looked specifically at Th17, Tc17, IL-17 producing gamma deltas, Th1, Tc1, GATA3 Tregs, and ROR gamma T Tregs.
Wow.
And
comprehensive.
Yes, and the gamma deltas were not as dramatically different as were some of the other T cell subsets. So they did not see the same difference in quite as dramatically in gamma deltas as they did.
I know they get seeded much earlier during development, so it could be that the hormones are playing more of a role later after.
Yeah, absolutely. And I could also, knowing what's coming in the paper, I can also guess another reason why that difference might be there. So because there's a change in T cell numbers in the skin of female mice, they looked to see whether the female mice made different responses to skin commensals or to skin pathogens. And they saw that they actually had higher T cell responses based on T cell numbers and based on bacterial load going down in response to both commensals and to pathogens. And they looked at the T cells, or they looked at keratinocytes. They saw big changes in things like antigen processing and presentation, things like that. So the next figure, or the next piece of this is sort of, I thought was pretty interesting, because they said, well, this could be that gene dose thing of differences in the number of X chromosomes, or this could be hormones. So how do we distinguish which of those things are leading to this difference? So they looked at mice before puberty, which they say is 3 to 4 weeks, and after puberty, which they say is 8 to 10 weeks.
Very fast.
Yes.
Saturation.
And the difference is only seen in the mature post-puberty mice. So the female mice before puberty don't have this increase in T cells, only the ones after puberty. They also took females and did ovarectomies, or took males and did castration. And the only place where they saw a difference were in the castrated males. And so the castrated males now had the increased number of T cells that females had. And so this also played out with the response to pathogen, the response to microbe, all of those things. But the idea was, okay, there's something going on here potentially with a male sex hormone. The male sex hormones are reducing T cells in the skin in these mice. And then they went on to what would not have been my first experiment, but that's because I've spent so much of my life thinking about T cells that they went to, well, what about the dendritic cells? Are there dendritic cells that are different in the skin that would be activating the T cells differently. And so, Cindy, part of this with your question about gamma deltas might be in relationship to how those gamma deltas are interacting with dendritic cells, because they next say, well, is the T cell difference due to a difference in dendritic cells? And so when they looked at dendritic cells, they saw that the females had more dendritic cells in the skin. So the percentages of individual subsets were the same, but the overall number of dendritic cells were higher in the females.
Interesting.
And the female dendritic cells had higher expression of genes associated with DC activation, DC differentiation, co-stimulation. The female DCs would migrate more. They were better able to activate T cells. And so these looked sort of like more mature, more activated, somewhat perhaps inflammatory dendritic cells in the skin of the female mice, which could, of course, lead to this increased number of activated T cells. And one of the things that they did that was really cool, I didn't even think of this caveat, but they did, and they addressed it, was they actually said, well, maybe the problem is actually that we're having a harder time getting the dendritic cells out of the skin. Maybe connective tissue is different, and so it's actually it's not an actual difference in dendritic cells, it's a difference in extraction. So they also figured out a way to fluorescently label them and look at them in real skin, so they didn't have to do extraction, to show that it was an actual difference in the cell number in the tissue, not just an extraction defect. And they have some pretty dramatic images here.
Yeah.
And, they do all of these experiments. The castrated males have the same change. Females treated with testosterone have reduced dendritic cells. And so it really looks like testosterone is reducing dendritic cell activation, which is leading to that T cell phenotype, specifically in the skin.
And your comment about the gamma deltas is because those dendritic cells would not typically be presenting to gamma delta T cells because they're recognizing different non-classical MHCs.
Exactly. So yeah, my thought is that maybe the gamma deltas aren't showing the same difference because this is coming through the dendritic cells.
I mean, the big question is, why would it be beneficial for females to have these extra T cells and more active dendritic cells in the skin?
Yeah, who knows? I don't know. And so they keep going with a mechanism.
Of a mouse, right?
Yeah,
because
from a mouse.
That's true.
The mice would have evolved in a much different environment than humans. So,
yeah.
So I think that my question is it directly the idea that it was evolutionarily beneficial to have these changes, or is it that this is a consequence of some other thing that was evolutionarily beneficial? Right?
Yeah.
And so then they kind of went to the next question of, well, which cell has the androgen receptor and which cell is getting signals from testosterone?
Yep.
And womp womp, skin dendritic cells don't have androgen receptor.
Oh, okay.
So that what they wanted was this to, be the androgen, the testosterone acting on the dendritic cell,
right,
and that changing the dendritic cell and thus changing the T cell.
Yeah.
So
D cells and T cells and dendritic cells talk to each other.
Right.
So if there's a change in the T cells, it could also affect the dendritic cells.
Right. So they decided to try to figure out which cells needed to have the androgen receptor. And they made a mouse that was missing androgen receptor in all lymphoid cells. Okay. And they found that it had to be a lymphoid cell making that was receiving the testosterone to get the signal. That in fact, if you had a mouse who had no androgen receptor in lymphoid cells, then the difference in dendritic cells was gone. So it was some lymphoid cell was impacting the dendritic cell. And when they went back to find what lymphoid cells in the skin have androgen receptor, they found that it was mostly ILC2s.
Okay.
So the innate lymphoid cells, which we don't talk a ton about I
No.
don't know that we've talked about them much. But these are cells that are coming from the lymphoid lineage, but that are not usually responding in kind of the traditional T cell receptor antigen dependent way. They're responding to more lymphoid like cues. An ILC2 in some ways has some parallels to a Th2 cell,
Yeah.
but is not necessarily responding with T cell receptor dependent signaling. And so they found that, in fact, ILC2s are increased in females only after puberty.
But is that the major subtype of T cell? I thought like all the different subtypes of T cells they looked at were up, right?
All those T cells were up, but the
Except
only
gamma
ones
deltas.
that seemed to respond to testosterone were the ILC2s.
Okay. So somehow they're also controlling the composition of the microenvironment as well.
Exactly. And so the idea is that the ILC2 is controlling the microenvironment. In a later part of the paper, they say that it's through their production of GM-CSF, through the production of another cytokine. That is altering the dendritic cell, and the dendritic cell is then altering the T cell. And in the end, the female mice are making much stronger responses to skin bacteria.
Interesting.
Compared to the male mice. And they do some adoptive transfers here to take a look at this. They also do a bunch of experiments. I guess I hadn't really thought about this before, where they used either RAG deficient mice, which we think of as being missing adaptive immune cells, and compared them to RAG deficient common gamma chain deficient mice, which they say are missing adaptive immune cells and ILCs, because they can't respond to the cytokines. And they could see that it was all of the numbers of dendritic cell changes, or all the dendritic cell changes, were due to ILCs. If you got rid of ILCs, you didn't see the change in dendritic cells. If you actually made ILCs that didn't have androgen receptor, you wouldn't have this change. And so all of this was due to the ILCs being signaled by testosterone that reduced the ILCs activity, reduced their ability to make GM-CSF. As a result, that made the dendritic cells worse. As a result, that reduced the T cell response. And so, sort of to your question, Cindy, my thought was, is it evolutionarily important for testosterone to impact ILCs? And the fact that it also impacts things way down the line is sort of collateral.
Right. And did they, I think I know the answer, but did they look at human skin T cells to see?
They did not look at human skin T cells here. No.
Okay.
That seems like something that could easily be done because there's lots of tissues available that you could stain or single cell data sets or whatever. Yeah.
But, maybe it's one of those things where it's like, this was a lot of work and it was very mechanistic, and they're like, this is a publishable unit. We don't need to have human stuff in here. They might have fought back on that.
Maybe. Yeah. Well, one of the reasons why I was really excited about reading this paper was that there were a couple of different talks I saw at AAI, conference we were at in Chicago, that really were tying some things, some endocrine responses to differences in immune responses in ways I hadn't expected. And so I remember more than once texting my colleague here, who is an endocrinologist, and telling her that I had a talk I needed to get her opinion on, and I needed to consult with her and learn some endocrinology. And so I think that this is just another place where we're seeing the fact that the immune cells don't just interact with themselves, but they're actually getting feedback from these other systems, like the endocrine system. And the obvious important idea that we should be looking at what's going on with immune responses in men and in women, and thinking about the role of hormones on all of these immune responses.
Totally. And thinking about the unique human cohorts that you could test these hormone questions. So, you can just look at males and females, but then you can look at transgender populations to understand if hormone treatment testosterone
Affects
particularly
anything.
affects
Right.
skin populations.
Yeah, or you could look before and after menopause, or
Mhm.
you could
Yep.
look at women who were on birth control pills. I think there are a lot of populations where these types of things might be interesting to analyze.
Right.
Yeah.
Right.
Cool.
Excellent. Okay, so the paper that I have to talk today, let me pull it up here. So the title is Maternal Diet Modulates the Infant Microbiome and Intestinal FLT3L, necessary for dendritic cell development and immunity to respiratory infection. So Brianne, I'm glad you did a nice, wonderful overview about dendritic cells because they play a big role in this paper. And the authors on this, the first author, I think single first author, is MD Al Amin Sikder, and the last author is Simon Phipps, and they are at the they're affiliated with the University of Queensland, but it also seems like they are a part of this QIMR. They may have a way of pronouncing that acronym, Berghofer Medical Research Institute in Brisbane, Australia. So, one of the things I thought I would do just to see what the audience thinks, if they like this way of presenting, they give a very nice overview of their findings. And I think Kathy Spindler has done this on TWIF before. So let's see if you all like this. But they're graphical abstracts. So I'm going to give you the overview of what they found, and then I'm going to go through the details and then come back to this overview and see if we're all on the same page. So what they found was that a maternal high-fiber diet alters the milk microbiome, and specifically the production of short-chain fatty acids that regulate the intestinal epithelial cell, and they highlight propionate as the specific short-chain fatty acid. We'll get into that. Modulates a particular receptor on the intestinal epithelial cell called FLT3L. It cleaves it off. FLT3L goes into circulation to alter the hematopoiesis of dendritic cells in the bone marrow. So it's like a soluble cytokine going from the gut to the blood to the bone marrow. And then those dendritic cells that move out of the bone marrow, they can provide host defense and disease resistance in the infant. Okay, so that's the overview. But let's then, I'm going to pull up my notes here. Where'd they go? Dive into the, how did they test this? What were the different things they did to prove that this was true? So the overview of why we care is, as we all know, the majority of childhood diseases, and particularly those that end up in the hospital, are lower respiratory tract infections that can lead to later complications like asthma. And they highlight those two diseases, both respiratory tract infections and later asthma, or other things like chronic obstructive pulmonary disease. They are all that having early life respiratory infections can lead to an increased risk of these things. And they are very prevalent in infancy. And it's very scary as a mom when your child comes down with a respiratory infection. And we ended up in the hospital with RSV last year with my 3-month-old. And this was before I really had access, which is a whole nother story about healthcare accessibility, but I had access to the RSV monoclonal antibodies that were on the market, but I just couldn't get them because my insurance company didn't deem my infant was high risk enough. But of course, we know that you could still have complications. So it's a big problem to try to understand how to prevent these infections, but particularly in resource poor areas or moms who have malnutrition. And when we talk about malnutrition, it could be, that you're not getting enough of a particular nutrient, that you don't have access to healthy diets. That would be high fiber diets like leafy green vegetables, or obesity, etc. So that's the context that they are investigating and trying to develop a preclinical model, their mouse model, to study the mechanisms by which this happens. And so what they had was a maternal low fiber diet and a maternal high fiber diet, and they looked at the pups and what they, how their responses were to a viral infection. And they use PVM, which is pneumonia virus, which is the same genus, pneumovirus, as RSV. So they're using this mouse virus as a model for RSV. And they found that while the weights of the mothers were not different or the pups at birth, it was post pneumonia virus infection that they found that the pups reared from low fiber diet fed mothers, they gained less weight, they had higher viral load in their airway epithelium, they produced lower amounts of interferon type 1, and all other hallmark features of a lower respiratory tract infection. This is higher neutrophils, epithelial sloughing, elevated pro-inflammatory cytokines, and mucus hyperproduction. So they really established their model that pups born to mothers with this low-fat diet they just did not fare as well to the infection. They also tested this with another virus, rhinovirus, which to us, we don't usually think of rhinovirus causing disease, but you could imagine in children, premature babies would be more immunocompromised and could have higher risk of symptomatic disease. So,
can I ask you something?
Yeah,
So
sure.
I'm trying to find it in here and I'm not seeing it. So were these mothers on this diet just during pregnancy or just after birth while they're nursing, or
I think it was throughout the time of conception, throughout lactation. So they did not do a preconceptus type of a study where they were looking at what the environment of the mother was before she conceived, but that is another potentially good study to do.
and did they nurse the pups on alternative moms that had the diet or not? Because could there be, and maybe you can answer this, could there be effects of the low-fat diet on the mom that's also affecting the fetus as it's developing? And this might not just be through milk?
So they did do cross-fostering studies, and
Okay.
they can reverse the severity of infection by taking low-fed diet pups and putting them on low fermentable fiber diet mice and putting them on the high-fiber diet. So at least in this context with this virus, of course, we always have to say that, this is mice, one virus, one diet. They did see that they could reverse the severity when they cross-fostered.
Okay.
So they talk about other birth cohorts, and specifically the asthma risk factor. And they tested this. So they took all the pups, and the ones who had the severe lower respiratory tract infection were all of the low fiber diet fed pups, and they treated all of them with this low dose cockroach extract. And this was a way to sensitize later in life after this challenge with the virus, who were more likely to have this characteristic a type 2 response. So this would be type 2 cytokines, an increase in lung eosinophils, serum IgE, all of the classic asthma responses. And they found that only the mice that had the lower respiratory infection in infancy, which was the ones fed the low fiber diet, had this asthma response. So again, they're really developing a nice phenotype for what we see clinically in infants and then children later in life. So their next question, well, how does a maternal low-fat diet have these impacts in the pups after infection? And they noticed that neonatal plasmacytoid dendritic cells and T regulatory cells were decreased. And they had previously shown that there was this crosstalk between dendritic cells and T regulatory cells that helped prevent immune-mediated tissue damage in this viral infection. They just weren't looking at it within this maternal diet and then the pup's context. So they took that. I think that's what led them. We think about why, like Brianne, you brought up that you would have gone one way, but they went to the dendritic cells. I think a lot of it is your previous data. How is this all leading you along the story? I mean, for us too, when we get a result, we're already kind of going down a train and I'm sure as in biology, there are many other stories that one could investigate, but they really wanted to understand the dendritic cell and the Treg story. And what they really thought was interesting is that they were finding that Treg cell numbers were higher in the mediastinal lymph nodes in these high-fat diets, but they found the same thing throughout the body. So it wasn't just in the lungs. It was in the bone marrow. It was in the gut and small intestine and colon. And they brought that up a couple times, that this is not restricted to the lungs. And so they were trying to, so this is a diet-induced response. So we're thinking, okay, the gut, but it's protected in the lungs. So that's leading us to thinking about the gut-lung axis. And so they found lower numbers of Tregs, they found lower numbers of dendritic cells, but they found lower numbers of many different subtypes of dendritic cells. It wasn't just the one subtype they were looking at, which led them to think that this was an early dendritic cell progenitor problem. So this is a hematopoiesis problem. And they did find that in the pups born to the low fiber diet mothers, they had lower levels of all major DC progenitor populations in the bone marrow. And so because they're going along the lines of this is a DC dendritic cell problem, let's do adoptive transfer studies to reverse the phenotype that they were seeing in their susceptible pups. So to implicate dendritic cell, and when I say PDC, that's the acronym, plasmacytoid dendritic cells, in conferring protection, they adoptively transferred purified dendritic cells from the pups born to the high fiber diet moms to the pups. So this is neonatal adoptive transfer. I can only imagine the dosing and how small the needles have to be. And these pups are very...
That sounds really hard.
Yeah.
It's whenever we start getting into like the neonatal models, it's just a different size dynamic to consider. But they were able to reverse the phenotype and found that the pups that received the dendritic cells from the protected mom, born to the high fiber diet, they were protected. And similarly,
Crazy.
They were able to reverse that phenotype. And when they did that, when they took the cells from the pups born to the low fiber diet and put them in the high fiber diet pups, they saw a decrease in those Tregs because the function of those cells was responsible for the Treg proliferation. So the adoptive transfer, again, was showing that dendritic cells matter. And they also did this in humanized mice. They did this in pups. So they're humanizing pups. They grafted CD34 positive hematopoietic stem cells into each of these pup populations, but reversed, of course. And they found that the human dendritic cells were numerically lower in the bone marrow and also in the lungs of the low fiber diet reared pups relative to the high fiber diet reared pups. So these experiments collectively are indicating that maternal intake of dietary fiber is positively affecting dendritic cells and Treg homeostasis, particularly in infancy. So that's a big jump. We're going from mom is eating something, and now the dendritic cells and Tregs in the bone marrow and throughout the body are affected. So what
In the baby.
the baby,
Not in the mom.
right? In the baby, not in the mom. So what's happening in between there? What is the reason why a mom's diet is affecting the baby so severely? And they found what I guess I could ask the team, what would you first look at if you're trying to, what maternal effect would you first look for?
Well, I asked about, is it, inherent to when she's pregnant or milk? And
Right.
You said they did do the studies to transfer,
They
put them
do
foster
that.
moms on different moms that had eaten high fiber or low fiber, regardless, if the pup had come from a mom that was eating chow or whatever. So something in the milk.
Right. It could be something in the milk or something else that the pup is exposed to in that cage, which is the feces of the mom.
No,
So you have
the
these two
microbiota.
things,
Yep,
the
yeah.
microbiota of
Okay.
the cage, which is the poop, your fecal microbiome, or the milk. Those are
Or is it's not something coming across the placenta?
correct. Based on the cross-fostering studies that they were to reverse the phenotype postnatally suggests that's not a placental, but it's a good point. So they tested the first thing, the fecal microbiome. They wanted to see if they could see this effect when they orally gavaged pups the feces from I always make this acronym wrong, high fiber diet moms. They orally gavaged the mother's feces to the susceptible low fiber diet pups to see if they could reduce the severity. And they did not.
How old
see
were they when they did this? Because if they're really, really tiny, gavaging is not so easy. It's hard to gavage a large mouse, let alone a tiny mouse.
I know. So when we gavage mice for our rotavirus studies, we can do you have to, so there's two different types of gavage. So you can do the ball, the little, it's a steel
ball one, yeah.
ball, or a tube, a syringe. And the syringe is a little bit gentler. And so we can do it as early as 4 to 5 days postpartum. And it's just
Wow, really?
very, 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 you do it enough and you get used to it.
Wow.
So you can do it as early as 3 days, even if you're really good.
They're really tiny.
They're so tiny. So mom's poop from a high fiber diet mom did not protect the low fiber diet pups from severity. So it wasn't that. Now they did the opposite, but the problem is they're still suckling on their mom. And so they were trying to induce disease in the high fiber diet pups, but they were suckling high fiber diet moms. So it didn't work out that way. But overall, they felt like, and I agree that the poop really wasn't the issue, but it was the composition of the milk. So what they did was first just look at correlations to the composition of the milk microbiome and the short chain fatty acids, which are produced from microbes. Did they differ in response to the variation in the maternal diet? And they did. Other things in milk like secretory IgA, epidermal growth factor, TGF beta, they did not change in relation to the maternal diet. It was the microbiome and the short chain fatty acids. So they were led down this path of milk microbes. So what they did was they enriched they sequenced the milk, and they took the dominant taxa and they tried to enrich for those taxa in an anaerobic chamber and then gave those to the pup. They tried to down select what is in the mom that could be causing this effect. And they did sequencing and they found Enterobacter, Enterococcus that are reported to be in milk. And they were able to show that those were able to protect the low fiber diet pups if they were given the microbes enriched from the milk of the mothers fed the high fiber diet. So it's definitely the microbes that are doing the business. And it seems that these microbes are enriched for propionate production. Now, this part of the study, there are other short chain fatty acids. There's butyrate and acetate. And I think this paper doesn't say that propionate is the only one that can do this, but it did show that it was the dominant.
I think that propionate has been shown to be particularly important in a lot of different sort of microbiome metabolism types of studies. So I don't think it's totally shocking to suddenly see propionate coming up again.
Right. Yes, agreed. And the interactions with these immune cell subset differentiation. Yeah, for sure. So do you say propionate?
That's how I learned to say it.
What do you say, Cindy?
Propionate.
Okay. I'm up for either, but sometimes I have caught myself, and I'm sure others have had this experience where you only have read a word in a certain field.
Yeah. Oh, yeah.
And then you go to the conference and you say it wrong. And everyone's looking at you. You're like, oh my God.
And that's where you have to learn. You should not make fun of people for pronouncing words wrong because they've only read things, because that means they were reading. Don't make fun of people for reading.
I know. Maybe you'll experience this. So my son used to read a lot, and he read obviously above his grade level, and so he was reading words that he had never seen before, and so he decided how they were pronounced in his head, just like you were saying, and he would use the word in conversation. And what are you saying? I don't understand that word. And then we would figure it out. But it would take him a long time to unlearn that, right? Because if you have never heard it before and you just figure it, and in science, when you start, I mean, everything is a foreign word pretty much.
Oh, for sure.
No, I have that in science and in non-science things. I still remember, reading the Harry Potter books before watching the movies. And Harry had a friend named Hermione.
Of course. How would you know to say Hermione?
Right.
And is that even how you pronounce it?
Yeah, it is.
I wasn't a Harry Potter kid, so I don't know.
Yeah, it is.
Okay. Yeah. So for anyone, you know what, forgive people for their mispronunciations and just be gentle with them, because it happens to all of us.
Tomato, tomato.
Oh my gosh. So they're, they then are giving propionate. Now I can't even say it the way that I originally said it. They're supplementing with propionate and giving it, and they're seeing a reversal in the severity of the disease, and that is associated with an increase in conventional dendritic cells, plasmacytoid dendritic cells, and Tregs in the lymph node. And so they then could reverse the effects they were seeing with the later in life type 2 development, IgE production, and airway remodeling. So it really was suggesting that this short chain fatty acid potentially driven from the microbes. And that middle part, I think, still needs fleshed out. And I think that's why the title of the paper did not say anything about milk or the specific short chain fatty acid, because I think there are more studies to be done there to really hash that out. But they then went on a dive and they said, okay, it's the propionate in the milk potentially, or at least supplementing from the microbes. And then how is it affecting the lung? So they found that the expression of this FLT3L, and this is the gene expression of this protein, has been shown to affect dendritic cell hematopoiesis. This is one of those where they said, oh, and now we looked at this, to me, obscure looking. And I'm just like, okay, I'm along for the ride, because I wouldn't have known. It was kind of just popped out of seemingly to me nowhere, but probably that was in their mind, maybe from their past experiments. And they found that the level of this protein was lower in the pups born to suckling on moms fed the low fiber diet. And it seemed that, the gene expression, this protein, it's expressed in different places: small intestine, colon, bone marrow. What they really hashed out here, that FLT3L was expressed on the surface of intestinal epithelium, but not in the bone. It wasn't expressed on the surface of cells in the bone marrow. It was really in the gut, able to act as a cytokine, because there's a particular enzyme that can cleave it off of the surface of the epithelial cell, and then it becomes soluble. And when you have something soluble, it can go into the bloodstream and act systemically. And this is where they really start to link what's happening in the gut to what's potentially happening in the bone marrow and then in the lung, because they blocked this particular cleavage enzyme called ADAM10. ADAM10 cleaves surface expression of FLT3L so that it can become soluble. They block that, and they see the ablation of the propionate-induced increases in all of these subsets. They see a decrease in protection from the virus. So it does seem to be that making this thing, the FLT3L, go soluble, go into the systemic, and then act externally outside of the gut, had the effect that they were seeing. And they found that this was very temporal. And what I mean by that is this wave of FLT. I have a hard time saying this fast.
I learned it as flat 3 like
I learned it as split
3 and flat 3.
Okay. Well, I have only, again, read it, so I'm glad you said that. So flat 3, or FLT3L.
Yeah.
Pick whatever one you want.
Whatever one you want.
Whatever one you want. But it was the expression. They call it a postnatal burst of FLT3. I like FLT3. It's a little bit easier for me to say. Affected by the fermentable fiber in the maternal diet that regulated neonatal development of plasmacytoid dendritic cells and Tregs, which protected them from virus infection. And that really wraps up. So if we think about where we started, there was a very strong phenotype with the mothers fed the different types of fermentable fiber diets, and the pups born to the low fiber diet moms, they were much more susceptible to disease. And it seems to be that in breast milk, there was a microbe that produced a short chain fatty acid that caused a burst of FLT3 expression, which could be cleaved by ADAM10. FLT3 went into the systemic and then promoted dendritic cell expansion outside of the intestine in the bone marrow, as well as Tregs, to then protect the lung. And I just thought it was a really well done paper. There's questions. I think, of course, as all, there's always questions. Could this be induced by another short chain fatty acid? Probably the answer might be yes. When I was doing some digging, in human milk, there's not as much data as we need, but I didn't find that there's really much propionate in milk. So it kind of made me wonder, if this is how this is working, do the bacteria have to actually be in the neonatal gut to start producing the propionate because if we're not detecting, see, they didn't measure it in milk, and that's what I would have made them do. I want to know what the propionate levels are in milk, and I can come over and teach y'all how to milk mice if you really want to figure that out. Because I just don't understand, like, the microbes are producing propionate, but where? And so could the milk itself do it?
Yeah, I would think it would be the microbes of the intestine of the pup
Yeah.
that would be producing the propionate.
I mean, they were able to take germ-free mice and treat them with propionate. So they could get the propionate from anywhere. It didn't have to be from a microbe that was in the gut. It could come supplemented.
But they never checked whether it was in the milk?
No.
And that seems like a crazy experience. That seems like a relatively easier thing to do. I mean, it's metabolomics. It's not super easy, but.
Right. I think maybe the hurdle is milking the mice. Or if you don't want to milk the mice, collecting the stomach blot, the milk blot, and you could try to measure it there, but maybe that's challenging because it's post-digestion.
Yeah, because you might also think that it might be the microbes and how they're digesting the milk oligosaccharide.
Yep. Totally. So I think that's why I didn't make it in the title of the paper, because that there's a lot of kind of
there's a big gap there.
Mm-hmm.
So I have a question, and that is FLT3 ligand is one of many, and I just Googled it, and there are probably 20 or 30 substrates for ADAM10, including Notch ligands, which are important for lymphocyte development at points. So how do they know it's truly the release of FLT3 that is the effect, rather than some other thing that ADAM10 cleaves?
So it's a good point. They bring that up in their limitations, that when they do an ADAM10 inhibitor, it can have effects far-reaching that are not just FLT3. They did not have, and they say this, they did not have a knockout animal that knocked out ADAM10 specifically in the intestine.
I think it's kind of important, because this could be all a true and unrelated thing as far as mechanism, right?
Right. Like it could be one of the many other
so
proteins that ADAM10
could be cleaved, but that is not the one that's doing the effect.
Right.
Yeah. I'm kind of wondering, they talk about, how important this is for respiratory infection. But if this is really going through, say, FLT3 or whatever we want it to go through, impacting systemic dendritic cells, then in theory, this should help those pups against a lot of different types of infections, not just respiratory infection.
Yep. Sure. Yeah. And maybe it was beyond the scope of the study to do rotavirus, to do like intestinal and others, but it's a great point. And yeah, so I think you're right, Cindy, a genetic mouse model that can conditionally knock out FLT3 in intestinal epithelial cells. And then, maybe trying some other model systems would be interesting.
It is nice that they put a limitations to the study thing at the end.
They do. They
right?
do.
We just talked about
that. We can appreciate that. So
yeah, and it does mention they need to conditionally delete FLT3 in the intestinal epithelial cells. So I guess, if you have such a provocative and new finding that a lot of other follow-up studies will, come with,
right.
I guess, yeah. But it would be a red herring if it's not actually FLT3, right? Because a lot of people could start going and looking at this, and it turns out that it's one of the other ADAM10 substrates. Yeah, a great point. And broadly, what I always think about with these maternal diet studies is like, when I was in my first trimester and vomiting every day, and I was not eating a high fiber diet. I was eating maybe like gummies, whatever I could like literally keep in my stomach. So my question would be, okay, if I'm really bad at having a high fiber diet for half my pregnancy, but then I get my act together because I can actually stomach things later, is that okay? What level of fiber do I need? When we translate this to humans, it's just much more dynamic type of questions.
Does it matter if you Is it more about what kind of fiber you have while you're breastfeeding? Does it matter how much fiber you had before you were pregnant? All of those things.
Right. And I guess to your point, if this is a milk, which they showed us, this is a milk specific phenotype, then what you eat postpartum. Yeah. Okay, so those are the 3 papers that we were able to go through. Anything else you want to promote, talk about?
No, I think that was it.
All right. Well, if that's the case, then that's Immune episode 80. Questions and comments can be sent to immune@microbe.tv. And like Cindy said at the beginning, consider supporting the science shows of Microbe TV at microbe.tv/contribute. Microbe TV is a 501c3. C3. So Cindy Leifer is at Cornell University at Cindy Leifer on Twitter.
Thank you.
Yes, you're welcome.
This is fun.
Yeah. Brianne Barker is at Drew University, and her handle is at BioProfBarker.
Thanks. I learned a lot.
Absolutely. And I'm Stephanie Langle. I'm at Case Western Reserve University, and I'm at Stephanie Langle. And you the music on Immune is by Steve Neal, and we really appreciate you listening to Immune, the podcast that is infectious. We'll be back next month.